Students, we are going to do quick revision of biology for the NEET exam within 30 minutes without wasting time. Some important topics we are going to cover. Parthenocarpy means plants which produce seedless fruits without fertilization. You can see the structure of microsporangium, TS of young anther, where the role of epidermis, endothecium, and middle layer is to protect the anther wall and it helps in dehiscence. Whereas tapetum, the inner layer, nourishes the developing pollen grain. Now, pollen grain, also called as male gamete, pollen grain also called as a male gamete. Its outer layer is called as exine and inner layer is called as intine. It has two cells, generative cell and vegetative cell. Outer layer exine is made up of sporopollenin, which is most resistant organic material, can withstand high temperature, strong acids and alkali, and there is no enzyme can degrade sporopollenin. Whereas intine is made up of cellulose and pectin. Now let us look at the diagram of anatropous ovule megasporangium, and here the funicle is the stalk of the ovule attached to the placenta, and raphe is the ridge formed by the fusion of funicle with ovule, and hilum is the junction between ovule and funicle. Now here you can see the embryo sac female gametophyte structure. It has 7 celled and 8 nucleate. Which are the 7 cells? 3 antipodals, 3 egg apparatus and 1 central cell. And central cell has 2 haploid nucleus, that's why it is 8 nucleate. It has 2 end, 2 opposite end, chalazal end and a micropylar end. And inside the synergids you can find filiform apparatus, and filiform apparatus main function is to guide the pollen tube. In artificial hybridization there are two techniques, emasculation and bagging. Another very important point to remember here is outbreeding devices. Outbreeding devices are the devices to prevent self-pollination, autogamy, and favors the cross-pollination. So which are the outbreeding devices? Unisexual flower, dichogamy means in bisexual flower, male and female parts matures at different time. Then one more is hercogamy. It's a physical barrier between anther and stigma. Heterostyly, where anther and stigma at different position, and self incompatibility. It inhibits the germination of pollen grains of same flower. So these are the outbreeding devices. Remember about cleistogamous flower and chasmogamous flower. Cleistogamous flower which do not open at all, and chasmogamous where you can find well exposed anthers and stigma. And false fruit is a fruit developed from ovary and thalamus. Example, you can find apple and cashew. And perisperm, you can find the remaining persistent nucellus here. Example, black pepper. And polyembryony means development of more than one embryo in a seed. Example, citrus. And during the formation of megaspore, that is embryo sac, you can find it is a monosporic development because only one functional megaspore develops into embryo sac. And apomixis is the plant produce seeds without fertilization, and pollen grains preserved in liquid nitrogen at minus 196 degrees Celsius. Oldest available seed is Lupinus arcticus, and the characteristics of insect pollinating flower, that is entomophyly, are the flowers are large and colorful, fragrant, rich in nectar, and safe place to lay the eggs. In TS of seminiferous tubule, remember the function of Sertoli cell. Sertoli cells nourishes the developing sperm, and there are some other cells called interstitial cell. You can find in the diagram are Leydig cells. Their function is to secrete male sex hormone, that is androgen, for example testosterone. Remember the organic connection between maternal tissue and fetus is placenta, and the hormones secreted by the placenta includes human chorionic gonadotropin, estrogen, human placental lactogen and progestogen. Menstrual cycle you can find in primates. It's a reproductive cycle. In female primate there are 4 phases: menstrual phase, follicular phase, ovulatory phase and luteal phase. Menstrual phase you can find breakdown of endometrium, inner layer of uterus, for 3 to 5 days. In follicular phase, you can find there is a secretion of follicle stimulating hormone and luteinizing hormone by pituitary gland. It occurs in 6th to 13th day of the cycle. In ovulatory phase, you can find LH surge. There is a rapid secretion of luteinizing hormone that helps in the rupturing of Graafian follicle to release the egg, that is occurs in 14th day. And luteal phase, ruptured Graafian follicle converts to corpus luteum, which is yellow in color, and secretes progesterone, which is to maintain the pregnancy, and this happens in 15 to 28th day. And remember, marula is a embryo with 8 to 16 blastomere. Seminal plasma contains fructose, calcium, and certain enzyme. And oxytocin is the hormone helps in parturition. And milk produced during initial days of lactation is colostrum rich in IgA. Fetal ejection reflex, it is originates from the fully developed fetus and placenta. And scrotum function is to maintain low temperature in the testes for spermatogenesis. Accessory ducts of male reproductive system includes retate testis, vasa efferentia, epididymis, and vas deferens. This order you must remember. And acrosome is a part of the sperm that helps in the entry of sperms into the ovum because it has some enzymes. And trophoblast is the outer layer of blastocyst. Next important topic is intrauterine devices. So the example you remember, non-medicated IUD is lip loop, copper releasing copper T and copper 7, and hormone releasing IUD is progestasert and LNG20. And remember the ideal features of contraceptives: user friendly, easily available, and no side effects, and it should be reversible. Natural contraceptive methods are three. It includes periodic abstinence, withdrawal method, and lactational amenorrhea. Remember, Saheli is once in a week pill developed by CDRI, and it's a oral contraceptive, and oral contraceptive are the pills made up of progesterone and estrogen hormones. Sexually transmitted diseases. Remember, in sexually transmitted diseases, gonorrhea, syphilis, genital worth, in that AIDS, genital herpes, and hepatitis B are the non-curable STDs, and Nirod is a popular brand of condom. Next very important topic is chromosomal theory of inheritance proposed by Sutan and Beveri. Here genes are located on the chromosome and no number of chromosomes is diploid in number. And the meaning of codominant, here dominant gene express their character without suppressing each other. Example, best example is AB blood group. And T.H. Morgan, he selected fruit fly Drosophila for its genetics experiment. The characteristics of why he selected fruit fly, because it completes the life cycle in 15 days, and single mating produce large number of progenies, and it shows sexual dimorphism. Linkage concept also developed by T.H. Morgan, where it is a physical association of genes on the chromosome. There are two types of linkage, complete and incomplete. And polygenic inheritance means traits controlled by 3 or more genes. Best example is skin color. And phenylketonuria also is the best example. Pleiotropy means single gene can exhibit many phenotypic characters. Example, phenylketonuria, starch in pea seed. Point mutation, only single base pair of the DNA is going to alter. Best example is sickle cell anemia. And multiple alleles where more than one allele controls the same character, ABO blood group. In Down syndrome, remember the karyotype, it is 45A plus XX or XY. Here you can find 21st chromosome will be extra. Features of Down syndrome patients are short-statured, furrowed tongue, and partially open mouth. In Turner syndrome, there will be an absence of one X chromosome, X0, where females are sterile and mentally retarded and broad chest. In Klinefelter syndrome, there is a presence of one extra X chromosome, males are sterile and develop gynecomastia. In the features of genetic code, remember here the genetic codes are triplet, they are universal, they are specific, they don't have any punctuations. And AUG, initiator codon AUG, plays a dual role. It codes for methionine also, and it will act as an initiator codon. And there are 3 codons, you can see UAA, UAG, and UGA. They are the terminator codons or stop codons. In Silent Futures of Human Genome Project, remember human genome contains 3164.7 billion nucleotides, and average gene contains 3,000 bases. And largest known gene, very important, dystrophin, and less than 2% of the genome codes for the protein. And chromosome 1 has most genes, and Y chromosome has less number of genes, only 231. And when you see the goals of Human Genome Project, it is to identify 20,000 to 25,000 genes in human, determines 3 million chemical bases, stores the database, improves the tools for analysis, and transfer to the related sectors. The next topic is in the structure of nucleosome and or packing of DNA. Remember, DNA wrapped around a positively charged histone octamer to form nucleosome, and histone rich in basic amino acid. Which are those basic amino acid? Lysine and arginine. And nucleosome contains 200 base pairs. Nucleosome further undergo condensation to form chromatin fiber and converts to chromosome. There are two types of chromatin, euchromatin and heterochromatin. And remember, euchromatin loosely packed, light stain and transcriptionally active, whereas heterochromatin is densely packed, dark stain and transcriptionally inactive. Next is, remember the division of labor of RNA polymerase. RNA polymerase 1 transcribes rRNA, 2 transcribes hnRNA, precursor of mRNA, and 3rd transcribes tRNA. Remember the 3 post-transcriptional modification in eukaryotes. It is a splicing. First one is splicing to remove the introns and joining the exons. Second is capping, addition of methyl guanosine triphosphate at 5' end of hnRNA. And tailing is addition of adenylate residues at 3' end of hnRNA. Remember the structure of transcription unit. Here the strand, first strand 3' to 5' is a template strand, and below you can find 5' to 3', it is a coding strand, and you can find two region, promoter region and terminator region. Between them you can find structural genes. Remember the full form of VNTR, variable number tandem repeat, and you can find N-glycosidic bond between pentose sugar and nitrogen base, very important, and phosphodiester bond you can find between two nucleotides. Central dogma of molecular biology developed by Francis Crick. It says that DNA converts to DNA by replication, DNA converts to RNA by transcription, and RNA converts to protein by translation. Very important. And Chargaff's rule says that in DNA, number of purines equals to number of pyrimidines. One more important point here you remember, DNA fingerprinting developed by Alec Jaffrey. Now the criteria that molecule can act as genetic material is, the genetic material should be replicate, structurally stable, and undergo slow mutation. In evolution chapter, remember, Miller experiment provides the proof of chemical evolution. During conducting this experiment, Miller used the composition of 4 gases, methane, ammonia, water vapors, and hydrogen. And after 8 days of working, he found amino acid in the liquid water trap. And he used 800 degrees Celsius temperature here. Hardy-Weinberg principle, also called as genetic equilibrium, where gene frequency in a population remains constant generation after generation unless it is altered by external agency. There are factors which affects Hardy-Weinberg principle like gene flow, mutation, genetic drift, genetic recombination, and natural selection. Remember the definition of saltation. It's a single step large mutation. Now you can find the definition and example of homologous organs. Homologous organs are the organs having similar structure and origin but performs different function. Example, forelimb of man, human, cheetah, bat, and whale. And in plants you can find thorns of bougainvillea and tendrils of cucurbita. And homologous organ is because of divergent evolution. Analogous organs are the organs having different structure and origin but similar function. Example, wings of butterfly and birds. In plants, sweet potato and potato. And analogous organs responsible because of convergent evolution. And adaptive radiation means evolution of different species in a given geographical area starting from a point and radiating to other area. Example, Darwin finches of Galapagos Island. In stages of human evolution, remember, Dryopithecus, they evolved 15 million years ago. They are ape-like. Ramapithecus also 15 million years ago, but they are man-like. Australopithecus 2 million years ago, they have 500 to 600 brain capacity. They usually ate fruit. And Homo habilis 650 to 800 cc brain capacity, not ate meat and use stone weapons. Homo erectus 1.5 billion years ago, 900 cc brain capacity, meat eater and lived in caves. Neanderthal man's 1400 cc brain capacity, hide to protect and buried the dead bodies. And Homo sapien 75,000 to 10,000 years ago, 1600 cc, they developed cave arts and they developed agriculture 10,000 years ago. The characteristics of each stage of human evolution is important to the exam. In the life cycle of HIV virus is a retrovirus because genetic material is RNA. Remember the function of enzyme restriction, reverse transcriptase enzyme that converts viral RNA into viral DNA. So viral DNA is produced by reverse transcription. And HIV virus are going to damage T helper cells, and macrophages are the factories of HIV. Related to malaria, you remember, it is caused by a protozoa, Plasmodium vivax. Sporozoids are the infectious form of Plasmodium which enters the human, and malarial parasite ruptures RBC and release a toxin hemozoin which cause fever and chill. In humans, they show asexual phase, and in mosquito, they show sexual phase. They require two hosts. And female Anopheles mosquito, it acts as a vector. Remember the example of primary lymphoid organs, bone marrow and thymus, and secondary lymphoid organs, spleen, lymph nodes, and Peyer's patch. Next, barriers of innate immunity also very important. There are four barriers: physical, example skin; physiological, acid in stomach; cellular barrier, macrophages; and cytokine barrier, interferons. Interferons are produced by viral infected cell to protect the neighboring cells. Some other important points of this chapter is colostrum is a yellowish milk secreted during initial days of lactation. IgA antibody is present in it. Typhoid is caused by Salmonella typhi. Its characteristics are symptoms are high fever, constipation, stomach ache and headache. Vidal test is a diagnostic test. Malaria is caused by Plasmodium vivax. Symptoms chill, high fever, recurring every 3 to 4 days. Amoebiasis caused by Entamoeba histolytica. Symptoms includes constipation, mucus and blood clot in stool and abdominal pain. Ascariasis is caused by Ascaris lumbricoides. Symptoms are internal bleeding, blockage in the internal passage, muscular pain and fever. Ringworm is caused by Trichophyton. Symptoms dry, scaly lesions on the skin, nails and scalp. And one more important point, AIDS is diagnosed by ELISA. Some important points of microbes in human welfare. Remember, lactic acid bacteria, lactobacillus, which converts milk into curd. It increases vitamin B12 in milk. And Saccharomyces cerevisiae, called as a baker's yeast. It helps to prepare bread. And toddy is a traditional drink of South India made by the fermentation of sap from palm tree. And Swiss cheese is produced by Propionum bacterium shermani. It has large holes because of the release of carbon dioxide. Microbes in industrial products, remember, antibiotic like penicillin produced by Penicillium notatum, citric acid by Aspergillus niger, acetic acid by Acetobacter aceti, butyric acid by Clostridium botulinum, lipase enzyme used in detergent formulation, pectinase and protease to purify fruit juices. And there are three bioactive molecules like cyclosporine A. It is used as an immunosuppressive agent produced by Trichoderma polysporum. Statin, its function is to blood cholesterol lowering agent produced by Monascus purpurae. Streptokinase is a clot buster to treat myocardial patient produced by Streptococcus. Remember the biogas plant structure. There you can find major composition of gas, methane, carbon dioxide and H2S. And methanobacterium are the bacteria present in the rumen of cattle. It produced due to the breakdown of cellulose polysaccharide. Next important point in this chapter is microbes as biocontrol or biological control of pest and disease. Dragonflies and ladybirds used to get rid of aphids and mosquitoes. Bacillus thuringiensis to control caterpillar and butterfly. Trichoderma to control many plant pathogen, and baculovirus of genus nucleopolyhedrovirus. It is an excellent species specific narrow spectrum insecticide. Next important point in this chapter is microbes as biofertilizer. Remember, rhizobium is present in the root nodules of leguminous plants to fix atmospheric nitrogen. And free living bacteria are there like azospirillum and azobacterium. They also enrich nitrogen content in the soil. And mycorrhiza belongs to genus Glumus. It absorbs phosphorus. And cyanobacteria like Anabena and Nostoc fix atmospheric nitrogen. And remember the definition of flocks, masses of bacteria and fungal filament to form mesh-like structure. In biotechnology, remember the steps of recombinant DNA technology, use of restriction endonuclease enzyme, DNA ligase, and transformation. And here, plasmid PBR322 diagram also very important. It has selectable markers like ampicillin resistant gene and tetracycline resistant gene, and ORI site for replication, and ROP gene produce a protein that helps in replication, very important. And first recombinant DNA was constructed in 1972 by Boyer and Cohen. Features of cloning vector, if you remember, it should have origin of replication, selectable markers, and cloning sites. Restriction Restriction enzymes are of two types, exonuclease and endonuclease. Exonuclease removes the nucleotides from the ends of the DNA, whereas endonuclease removes the nucleotides at a specific position in the middle of the DNA. While seeing the convention for naming of restriction enzyme, for example, E. coli, first letter E is genus, then second two letters CO, for example, is from species, next letter represent the strain, and Roman number indicates the order of discovery. Agarose gel electrophoresis is the technique to separate the DNA fragments you can find, and to identify the separated DNA fragments there is a use of stain called ethidium bromide by exposing to UV light. And tools of recombinant DNA technology are restriction enzymes, DNA ligase, polymerase enzyme, vector or DNA plasmid, host cell, and bioreactor. Three steps of PCR you remember: polymerase chain reaction, denaturation, annealing, and extension. In this technique, there is a use of one specific enzyme called tag DNA polymerase obtained from Thermus aquaticus bacteria that withstand high temperature. In case of BT cotton, remember it has a insecticidal property. Bacillus thuringiensis produce one crystal protein that kills insects. And Bacillus thuringiensis has some genes like Cry1AC and Cry2AB to control cotton bollworm, and Cry1AB to control the corn borer. And this crystal protein exists in inactive protoxin but get converted into active toxin in the gut of the insect due to alkaline pH and create pores in the midgut and kills the insect. And remember the use of genetically modified organisms, GMO, to make the crop more tolerant to abiotic stresses, reduce dependency on chemical pesticides, reduce post-harvesting losses, and enhance nutritional value. And in the production of human insulin by genetic engineering, remember here the insulin is the first recombinant product has been developed. It has two chains, A chain and B chain. Insulin first produced in 1983 by Eli Lilly company. It's an American company to produce insulin. In the benefits of creating transgenic animal, remember some points like alpha-1 antitrypsin is used to treat emphysema, and first transgenic animal is cow Rosie. It produces a milk which is rich in alpha-1 antitrypsin. In organism and population, remember there are four levels of biological organization: organism, population, communities, and biomes. So, since tiger census is done by pug marks and fecal pellet, and to find out the population density, we have a formula. You can find NT plus 1 is equal to NT plus B plus I. B means birth rate, I means immigration, minus D plus E. D means death rate, and E means immigration. You can find two graphs here. One is exponential growth curve. It is a J shape where when it will happen when the resources are unlimited. Formula is dn by dt is equal to rn. R is extrinsic factor. And sigmoid curve is a logistic growth curve. Here you can find K, which is a carrying capacity. Now in predation, remember, there are some defense mechanism developed by prey against predator, like insects and frog are camouflage, cryptically colored. Monarch butterfly is highly distasteful, and you can find thorns in cactus, and calotropis produce a poisonous cardiac glycoside. One more important point here is resource partitioning. Means if two species compete for the same resource, they could avoid competition by choosing different times for feeding. Best example given by MacArthur: 5 related species of warblers living on the same tree. Remember about parasitic adaptations: loss of unnecessary sense organ, presence of sucker, and high reproductive capacity. Ectoparasite means example lice on humans, ticks on dogs, cuscuta plant grow on hedged plant. Endoparasite lives inside the host. Brood parasitism, where parasitic bird lay its egg in the nest of its host and let the host incubate them. Example, cuckoo lays egg in the nest of crow. In commensalism, you can remember that in this interaction, one species is benefits and other neither benefited not harmed. Example, orchid grow as epiphyte on mango tree. Another example, clownfish on tentacles of sea anemone. Next, barnacles on the back of whale. And next is cattle, egret and grazing cattle. Mutualism, in this interaction, both the organisms are equally benefited. Example, lichens, association of phycobiont algae and mycobiont fungi. Mycorrhiza, association of fungi and roots of higher plants. Pollinating insects and flowering plants. And fig tree and wasp. And sexual deceit you can find in orchid aphrys, where flower petals resembles the female bee and male bee attracted and responsible for pseudocopulation. Here you can find the pyramid of number with example, pyramid of biomass, remember with unit, and remember pyramid of energy is always upright, and some definitions are there like standing crop, 10% law, stratification, food web and food chain and its type are very important to the exam. In the chapter biodiversity and conservation, remember the causes of biodiversity loss, evil quartet. They includes 4, habitat loss and fragmentation. It's a main one. Over exploitation, for example, steelar cow and passenger pigeon. Co-extinction, for example, plant pollinator and host parasite. And alien species invasion, for example, Nile perch introduced into Lake Victoria causes extinction of cichlid fishes. And in the conservation, remember two types: in situ conservation and ex situ conservation. In situ conservation, conservation of endangered species in their own habitat, example biospheres and national parks. And ex situ conservation, conservation of endangered species in artificial habitat, for example botanical gardens and zoological park. Biodiversity is a sum total of species riches. It has 3 types: genetic, species and ecological. And Paul Ehrlich proposed revert popper hypothesis. Rauwolfia omitoria produce reserpine, example for genetic diversity. And Alexander von Humboldt, he shows species area relationship. And recent extinct as per IUCN, Dodo, Quagga, Steelar cow, 3 subspecies of tiger, Bali, Java and Caspian. And remember, sacred groups means these are the traditionally conserved forest and wildlife with care and protection. Example, Aravalli Hills in Rajasthan and Western Ghats of Karnataka. And endemism means these species are confined to one particular area and not found elsewhere.